Okay, great. Thank you, Thijs.
So good afternoon, everybody. Thanks for inviting me to this workshop. I'm pleased to take part. In the next 10 minutes, I'll give you some brief overview of the recent changes in the epidemiology of this Goose Guangdong lineage of high-path avian influenza virus that Michelle introduced. First, to continue on the Influenza 101 that Michelle started, I just want to give you a little bit of relevant information about this Goose Guangdong virus infection in wild birds. I have 4 main points, and there are references to show you more information about those, but you can also ask me about them later on if you're interested. So it's important to know that this virus can, in principle, infect any wild bird species or domestic bird species, but the disease that's associated with the infection can vary strongly. So while some species can be infected and not show any clinical signs at all, other species can be infected and show high mortality. The second important point I want to make is that influenza virus in birds is an acute infection. It means that infected birds stop excreting infectious virus after about 1 week. There are no known chronic shedders of influenza virus. The third thing is that there's a strong effect of immunity. That means that wild birds that have survived a past infection can be protected completely or partially from a new infection with the same variant of the virus. And we've carried some infections in the laboratory showing that even after 1 year, a previous infection will protect wild ducks from both excreting virus and from becoming ill from a virus. And the last thing I want to make is that the virus spreads not only from bird to bird, but that it's critical for the epidemiology of influenza that infection takes place via contaminated water, and for birds that eat other birds, either raptors or scavenging birds, via contaminated carcasses. So now to go to the epidemiology of this Goose Guangdong lineage. This Goose Guangdong lineage emerged in a commercial poultry farm, and that is the case with most of these emergence events of high-path avian influenza. They typically occur in intensive poultry production systems. And because the poultry sector has increased dramatically in the last few decades, and if business continues as usual, it will continue to increase dramatically in the coming decades, it's expected that the number of high-path avian influenza outbreaks in poultry will increase also, which means that if we want to prevent future outbreaks and emergence events in poultry, but also in wildlife and also in humans, it's the poultry sector that we really have to look for to prevent these emergence events. So going now specifically to this Goose Guangdong epidemiology, after it emerged in 1996, it remained limited to Asia until about 2004 and spread mainly through contaminated poultry products and infected poultry. So this is the typical thing for an outbreak of high-path avian influenza virus in areas where it's not so well controlled. But what happened subsequently was quite unusual. In the summer of 2005, the virus was suddenly found in Qinghai Lake in Western China, and a few months later had spread thousands of kilometers west to Europe, to the Middle East, and to Africa. So it had made a big expansion in its range, both infecting wild birds and infecting poultry. And we have in time come to learn that this range expansion occurred through migratory wild birds in a two-step process. In the first step, the virus was carried via spring migratory birds from wintering areas, for example, in South Korea, to the breeding area in northern Russia, Siberia, where it remained present during the breeding season. And in the subsequent autumn migration, while some birds returned with the virus to the original wintering areas, other wild birds from that breeding area moved with the virus to other wintering areas, for example, in Europe, and in 2014, 2015, also across the Bering Strait into North America. So this virus is able to spread via migratory birds over a long distance in a two-step process. And if we look over the years, after the original eruption in 2005, 2006, things remained quiet with regard to this long distance expansion until 2014, 2015, where the virus in a slightly different form spread not only to Europe, Africa, but also to North America. Then it was quiet for 1 year, and then every year after that there have been eruptions of the virus originating from Asia, going to Europe and Africa. And in the last 2 years, things have changed again, because in the last 2 years, the virus has adapted to maintenance in wild bird populations so that it can remain present in wild bird populations in one area all year round. And in the last year in Europe, we've had the largest high-path avian influenza virus outbreak ever recorded, with nearly 2,500 outbreaks in poultry farms, causing nearly 50 million poultry birds to be culled in Europe. There have been nearly 3,000 high-path avian influenza virus events recorded in wild birds. But an important point to make here is that all the AI surveillance for high-path avian influenza virus in Europe, but also in North America, is directed at protecting the poultry sector from incursion of the virus. And for this reason, there is interest to determine whether the virus is present in wild birds, but there's no specific interest to understand the actual scale of mortality in wild birds. So counting the number of wild birds is not part of the current European Union or North American AI surveillance effort. So this is a really important point to make for this group. So what was special in the last 2 years is that the virus has become maintained in wild bird populations occurring locally. And probably as a consequence of this, there has been frequent detection of the virus in the spring months of 2021 and 2022, whereas this never occurred in previous years. And this has allowed the virus to spread during spring to seabird colonies, causing high mortality. And not only that, it has also spread to juveniles of the year. So juvenile raptors, such as sea eagles, have died from the virus. Juvenile wild mammals, such as red foxes, have died of the virus. And because the virus is likely being present in spring migratory birds in Europe, the virus has also been able to spread across the Atlantic to North America. So this is a new event. North America had been infected with this virus once before, in 2014, 2015, as I explained. There, the virus had come from the other direction, from the Pacific Ocean across the Bering Strait into North America. But last year, the virus spread from Europe across the Atlantic Ocean, probably using either Iceland or Greenland as stepping stones, to North America, where it was first detected in St. John's in November 2021. And by phylogenetic analysis, we can see that the virus that was found in Newfoundland had the closest relationship to viruses that were found in Europe in the spring and early summer of 2021. Again, showing the importance of this frequent presence of the virus in the spring. So in conclusion, I think we have to really make a shift in our idea of high-path avian influenza. So rather than being unaffected vectors of spread, wild birds are rather hapless victims of this goose guangdong virus, with mortality levels that are of potential concern to these wild birds at the population level. Because we have international responsibilities regarding migratory bird populations, it should stimulate national authorities to prevent future high-path avian influenza outbreaks, not only in poultry and humans, but also in wild birds. And for this audience, I think it's important to note that it's important not only to take appropriate measures to reduce virus spread in seabird colonies and elsewhere where many wild birds aggregate, but also to register the number of wild birds that are found dead and to collect appropriate samples for virological analysis as factual evidence for policymakers to make decisions about preventing high-path AI in the future. So I'd like to acknowledge Horizon 2020, which are funding two projects that I'm involved in, Delta Flu and VEO, and also the members of the EFSA AI Working Group and members of the AI Impact Working Group, with whom I've had many discussions about avian influenza in wild birds, poultry, wild mammals, and humans. Thank you.
Thank you, Thijs. That was very, very interesting. We've got time for a couple of short questions if anyone in the audience has got a question. I have a short question, Thijs, whilst everyone else is thinking. Good to see you again. Mm-hmm. You mentioned that infections in wild birds are acute and last for just a week. Is there any suspicion that birds might be carriers and asymptomatic, or that is, what is the certainty about that they are acute and just 1 week?
Yeah, the certainty is high. Avian influenza virus has the, you could call it a hit and run strategy. So it has not evolved to deal with the specific immune response of the host. So it infects a bird, rapidly increases the number of virion particles that are produced. They are excreted, and once the specific immune response of the infected bird kicks in, the virus infection is abrogated. There's one area where the virus can remain for a little bit longer because it's an immune privileged site in the central nervous system. And in birds that have been able to survive the acute phase of the infection, they can still develop neurological signs and become ill from that. And that's what we see often with this particular lineage of virus, that animals are dying with neurologic signs of disease. But there's no evidence of a long-term infection in wild birds. There's one factor or fact that confuses some people, is that you can detect evidence of infection by PCR in, for example, cloacal swabs or pharyngeal swabs. So what you're detecting there is pieces of the genome of the virus, the RNA, but this is not infectious virus. So when you carry out parallel tests, testing for the presence of infectious virus and the presence of virus by PCR, it invariably, when you test for infectious virus, the virus is in the experiments that I've been involved in, usually 5 days after inoculation, the animals already have stopped excreting infectious virus. And in some other cases I know of, that it may last until a week. On very rare occasions, maybe 10 days or 2 weeks, but that's very rare already.
Okay, thank you, Thijs. That's very interesting. I'm sure lots of people will be bearing that in mind. Amandine has a question online, and then there's a question in the room. We've got a little bit more time for questions, if that's okay with you, Thijs, because obviously Michelle didn't have any questions to answer. But, Amandine.
Yeah, I'm actually asking, there are lots of questions that were asked before the workshop that I think you're the person who can actually maybe have an answer.
Okay.
One of the question is there a way to develop a rapid test similar to what we have for COVID, for instance, so we don't have to ship birds or samples?
Yes, there is a rapid antigen test, and there have actually been some trials with this rapid antigen test in rehabilitation centers in the Netherlands, and I think that that's a possibility for future use. Okay,
so it's not ready to be deployed yet, but it's potentially.
Well, I think it's been tested once. I don't know if you think that it's then ready to be deployed, but it looks like it works. Okay. But it's I think that the outcome of that first study is still ongoing.
Okay, great. Another question is, I mean, you mentioned the role of poultry, and one of the questions that everyone is asking is, can we do anything to prevent future outbreaks in the wild? And so I don't know if you see anything that we as seabirders could do, like besides. So I know there's a vaccination pass on one side. I don't know if you see any other things we could do.
Yeah, so for this particular event, I think it's already out of our hands. So the virus has escaped has spilled over into wild birds, this high path virus, and we've left it so long that it's adapted to wild birds, and now apparently can be maintained in wild birds. So there are several examples of such diseases coming from livestock that have spilled over into wild animals, such as African swine fever from domestic swine into wild boar, bovine tuberculosis. So there are several infections like this. So they don't occur very often, but they can have dramatic effects. And that's this virus probably, I speculate that this virus will remain in wild bird populations. What we can do as a society, but I think it's not just up to the seabirders, but as a society, is to really fundamentally change the way that we keep poultry, in order that the risk of another high path avian influenza emergence like this is strongly reduced. So that means not having such big poultry farms, not having poultry farms so close together, strongly improving our, let's say, veterinary care of poultry, so that the outbreaks are stopped as soon as possible. for example, by vaccinating them, things like that. So there is a possibility for preventing future high-path avian influenza outbreaks to happen again like this, but this current one we've let slip out of our hands.
And you mentioned maintenance in the seabirds, and there is one question in the chat, I think, kind of related to that is, do we know anything of the how much immunity there is? Like if the birds have already been exposed now, are they going to be protected against maybe infection or death?
Well, have to be a little bit careful because maybe every species maybe is a little bit different, but looking across the board, across the species, it looks like once birds have been infected with a virus and they have survived it, in the subsequent year, they will be less susceptible to either becoming infected with the virus, or if they do become infected, they will excrete less and they will also become less ill. And there's some very nice studies by Oliver Pybus and his colleagues in mute swans in the southern England, where they've looked at a mute swan population over the years, and they've shown very nicely that these mute swans have developed immunity, and when there's a new outbreak of high-path avian influenza, it's the juveniles that suffer mortality and morbidity, but the adult birds do not.
Okay.
So in that sense, you could speculate that on the longer term, we might expect that this virus will, if it remains present in seabird populations, and we also don't know that if it's going to stay in a smaller group of species, maybe more the enzeriform waterbirds, or still expand into seabirds. But if it does still occur in seabirds, then it would be mostly the young of the year that would suffer mortality, and not so much the older birds.
Okay. There is one last question in the chat. You mentioned the role of transmission through water. Do we know if it's the case also for salt water?
Yeah, that's an interesting question. So there have been many studies on the persistence and remaining infectiousness of different avian influenza variants in water, fresh water and salt water, cold water and warm water. And indeed, avian influenza virus can remain infectious in salt water for quite some time. Yes.
Okay.
So certainly for a number of days, maybe even weeks, in cold salt water, it can remain infectious. A point there is that talking about seabird colonies, where there is seawater, there's also a lot of current. So then the virus can be very quickly diluted. But where there's seawater that is in a stagnant pool, that's a different situation, and that might indeed be a source of contamination of other birds.
Okay, thank you. I give it back to you, Matt, if you have a question in the room.
Okay, yeah, there was one question in the room, Matty.
Yeah.
There's one on somebody's phone at the back as well, but we'll leave that one for now. Matty.
You said about the infection from carcasses could be important for virus spread, and we had lots of debates about this. Should we be collecting carcasses? Shouldn't we? Generally, government vets told us no. Should we be collecting carcasses to prevent spread, or is it already in the colony and therefore it won't make any difference?
Did you hear the question, Thijs?
I think I heard the question, yes. So that's a really good question. What to do when you have a mortality in a natural area, let's say at a colony site. Do you go into the colony site, disturb the birds with the potential of spreading the virus further to collect the carcasses, or you just leave them there? I think you have to take it on a site-by-site basis. You have to make that decision on a side-by-side basis, and you have to make that decision together with people who have experience at that site with the species involved, because some species are much more likely to maybe abandon a site by such disturbance than other species. However, it is so that letting the carcasses remain on site means that those carcasses can remain infectious for in cool weather for maybe several weeks, or maybe even longer, and remain infectious for other birds. So they're remaining a source of contamination for other birds. Great skuas, I have read, will feed on carcasses of their own species. So in that way, they may promulgate the epidemic in their colony. Sandwich terns actually will try to mate with dead sandwich tern carcasses, necrophilia. So that's also a very good way of spreading the infection. So I think that, again, on a side-by-side basis, you really have to think seriously about collecting the carcasses to reduce the risk or reduce the spread of the virus and reduce the impact of the epidemic on the breeding colony.
Okay, thank you, Thijs. Lots of people were writing furiously in the answers to your questions. So thank you very much for your time there. Thank you, Thijs. Very interesting stuff.